Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we are talking about techniques used in cell biology. As you know, cell is a microscopic structure. Therefore, for detailed study of cell, scientists have developed different techniques. These techniques include microscopy, fractionation, chromatography, staining, microdissection, micrometry, etc. In this video lecture, we will discuss one of these techniques, and that is cell fractionation technique. Now, what is the objective of studying these techniques? So, the objective of studying these techniques are: number one, to realize the great efforts of the scientist who has made things easy for us. And number two, to understand the purpose of using these techniques so that we can also use them when required. Now, what is the definition of cell friction technique? It is a process used to separate several components without altering their structure and function. Now, this is important, that the process used to separate several components without altering their structures and functions. Now, how is it possible? Procedure of cell fractionation technique. In fact, there are two main steps of cell fractionation process. The first one is homogenization, while the second one is centrifugation. The homogenization is a process whereby cell or tissue is ground in a machine called homogenizer or food mixture in a suitable medium with correct pH and temperature. So homogenization, as I told you, is the process in which cell components are separated from one another. Now, you have to keep in your mind that cell components are not separated from the cell. For example, you see this is the cell, and in this cell these are different organelles of the cell. These cell organelles are not separated from one another completely. For example, you can see endoplasmic reticulum is attached with nucleus, and Golgi bodies may attach with endoplasmic reticulum, and ribosomes are attached with endoplasmic reticulum, and endoplasmic reticulum may also attach with cell membrane. So the purpose of homogenization is to separate these cell organelles from one another so that they can be centrifuged easily. So for homogenization process, two things are important. The first is the correct pH. So it means that we have to keep the cell tissues in a buffer solution, while temperature is also important, because high temperature can denature the enzyme activity and low temperature can stop the enzyme activity. Therefore, we want to stop the enzyme activity in this case. Therefore, low temperature for homogenization process is required. So, the other step after homogenization, the substance in the test tube is called homogenate. This homogenate is then shifted to a machine which is called centrifuge machine, and the process of separation of cell organelles in centrifuge machine is called centrifugation. In fact, centrifugation is the technique which involves the application of centrifugal force to separate particles from a solution according to shape, size, density, speed of rotator, etc. So here you can see the centrifuge machine, and in this centrifuge machine there is a place for test tube, and this is in fact an automated machine, and it can be adjusted according to requirement of the procedure. And there is a homogenizer. So the process of centrifugation can be understood by this diagram. In this diagram you can see that there is a test tube with homogenate, and when we keep this test tube in centrifuge at low speed, we find after a specific time that at the bottom of the tube some algaes has been settled down, and these settled algaes in the bottom of the test tube are called pellet, while the remaining suspended portion of the homogenate in test tube is called supernatant. Now, usually in this pellet we can find large size algaes, because in low speed only large size algaes can settle down. Now, if we continue the process to separate the other algaes which are smaller than these, then we will shift the supernatant into another empty tube and then repeat the process in centrifuge, and we will find another pellet in this tube and supernatant. In this case we used medium speed to separate algaes which are smaller than nucleus. For example, here we will usually find the mitochondria, lysosomes, etc. And if we further want to continue the process and for the separation of even smaller algaes, then we will shift the supernatant into another tube and again centrifuge this tube with high speed in this time, because high speed is required for the centrifugation of smaller algaes. So here we will find in the pellet, and in this pellet usually large sized molecule or cell algaes will be found, like ribosomes, etc. Ribosomes, etc. Now, by this discussion, we can find that for larger molecules, less speed is needed, like nucleus or cytosomes, and for smaller molecules, more speed is needed, like ribosomes, viruses, and large macromolecules. So this is the sequence of sedimentation. So after studying the procedure of centrifugation, another thing which students have to keep in their mind, that there are two different types of centrifugation. The first type is called the differential centrifugation. It is called differential centrifugation because it differentiates smaller and larger particles from each other. But this is not accurate for specific algaes separation, because the two algaes may be of the same size. The other procedure is called density gradient centrifugation. It is called density gradient centrifugation because it separates components on the basis of density in a density gradient medium. So it is more accurate process because this process can separate specific organelles from each other. So these are the two types of centrifugation. And at the end, we should also know this fact that the centrifugation process is not used only in cell biology, but this process is very commonly used in diagnostic labs to determine and to identify the different causes of diseases, especially pathogenic diseases. And this certification process is also followed in forensic science to determine the criminal cases, as well as in microbiology labs and biochemistry labs for different purposes. So that's the end of this lecture. Hope that this lecture will be fruitful for you.